let me begin by introducing myself. I am Vibha Lade, a member of the marketing and outreach team of Eureka, which is an online scientific newsletter run by the students of NMIMS, Mumbai School of Science. And for February's issue, it is around genetics and epigenetics. So today we need your expertise to guide us. So,
Okay.
could you give us a short like an introduction of yourself?
So my name is Sohni Tagore, and I'm currently working as a research analyst at the GeneBox, Mumbai, wherein I specialize and focus in clinical genomics, pharmacogenomics, and also epigenetics in terms of product development. So my career history is basically something like I did my bachelor's from DY Patil, Navi Mumbai School of Biotechnology. Thereon, I went to get my master's in medical genetics and genomics from University of Glasgow on a scholarship. And then I completed a 1.5 year research stint at the University of Antwerp in conjunction with USA, that is the University Hospital for Antwerp, in the area of hereditary hearing loss. And then because of COVID, I had to come back and start all over again in Mumbai. So yeah, that's my kind of short introduction for myself.
Uh, okay, so along with you, we also have our editors here today for introduction of yourself. Maybe Sanya can start.
So, hi, I am Sanya Damle. So, currently, I am in my third year of biomedical sciences, somehow figuring out the way through it, one step at a time, one day at a time. But then, yeah, I think epigenetics is something which interests me. So, I was like, okay, I'm going to be there for this and I want to hear like the insights of it. So, yeah, Harsh,
Okay, hi everyone. I am Harsh Bokadia and I'm in the fourth year of my integrated biomedical science course. So basically first year master's. And I think with a lot of fields focusing more on genetics, like NGS, which is a very recent and hot topic right now. I think the theme for this issue, that is genetics and epigenetics, is really relevant to the time and to have you as our expert interviewee. I think we are very honored to have you here. So yes, very excited for the interview.
Thank you so much for having me.
Okay, so to begin with the interview, just to give you a brief, today's interview has two segments. So the first segment has the interview questions, like asking you what it is and everything. And the second segment, it is a kind of a fun segment asking you like whether it's a myth or a fact. We'll be giving you the statement, and then you
Yes.
can say if it's a myth or a fact.
Okay.
So let's begin with our first segment. And the first question that we have for you today is, could you share what inspired your journey into the field of genetics and how your undergraduate work in cytogenetic analysis help shape your career?
Okay. So when I was getting into the selection of my bachelor's, I knew for sure that I wanted to do genetics only. Like वो बहुत सारे बच्चे कहते हैं ना कि MBBS नहीं मिला तो आ गए बायोटेक में, वैसे नहीं था मेरा. When I did my 12th standard का project for biology, I was very inspired by my biology teacher, and I did a project on blood groups in MGM hospital. And I was like, this is what I'm going to do. This is what I'm going to focus on. So therefore, when I jumped into my degree, I was very curious about how, cytogenetics works, how genetics works. And since my childhood, I had been facing all of that. I'm the only one in my family who has curly hair. Everyone in my family has straight hair. Why do I have curly hair? How do I manage this? I'm the only one with the O positive blood group. Why does everyone in my family have B positive blood group? That was very intriguing to me. So I focused on that, and then I went ahead. So उसकी वजह से जो passion आया है, and I'm like, no one can get me into genetics as much as my passion can. So I followed in my footsteps and treaded on it. And then I had a very good teacher for my bachelor's animal tissue culture, Dr. Mala Parab, and she guided me that there is an upcoming field for cytogenetics, genetic counseling. And I know you are very empathetic. You can go and do something in there. So she referred me to MGM Hospital Vashi, and there I met Dr. Bani Ganguly. She interviewed me, and she was like, "Sure, you can do your project here." And thereafter, what happened in those 6 months of my bachelor's thesis was I was constantly given samples for patients with Down syndrome, autism spectrum disorder. So that kind of, gave me an analytical view into how broad this field is, not just fish and karyotyping. You also need to counsel with the patients. You know what genetic counseling actually is. How do you draw the pedigree charts? How do you talk to the patients? How do you counsel the families? And I was so like the psychological and emotional toll at that age. You at what, 18, 19 years? You don't know how to take it. So I was like, I need to understand more. So I simultaneously took permission from my college authorities and I was like, I'll do a psychological counseling course also, side by side, from graduate of Indian Institute of Counseling, Delhi. So I did that, and then that helped me grasp a bit more on how to talk to these families in a better way. And I was not talking in the hospital. I was under my guidance going to another hospital for underprivileged children in Govandi. So patients were treated free over there. So the education level of those families was not so high. हमें genetics के concept explain करने में normal लोगों को दिक्कत हो जाती है. Here I was trying to explain these concepts to people who are only able to speak in Marathi or Hindi, or they've migrated from other parts of India and then they are, here for free services because it's so expensive genetic testing, as we all know. So because that was funded by the hospital, MGM Hospital, as a part of their outreach program, I could go and talk to these patients. Obviously, there was 2, 3 other students with me. We were doing a group project, but it was an eye-opening experience how to, go ahead in genetics. Cytogenetics is just a part of it. You can do so much more. So that was how I came into cytogenetics and, all of this. So I hope that answers your question.
It does. So when we were doing like a search of your LinkedIn and everything, we found that your master's research, it focused
Yeah.
on gene regulation and glioblastoma. So
Yeah.
what challenges did you encounter while investigating the therapeutic targets, and how did this experience expertise, how did this enhance your expertise in bioinformatics?
Yeah. So after I completed my BTech thesis, I was like, I'm not ready for this emotional burden of being a genetic counselor. That's not my forte. So I chose to go for my medical genetics degree because, I could be behind the scenes and still make an impact. So my master's thesis actually for glioblastoma multiforme, GBM, which is a very aggressive form of brain tumor and has a very not a very good prognosis rate, like 12 to 18 months post diagnosis. So I was like, I'll focus on this. And there, my guide, Dr. Catherine West, was dealing in all of this, like glioblastoma multiforme and RNA-seq analysis at that point of time. This is 2019 I'm talking about. So I was very intrigued by that. Wow, RNA-seq analysis, we are not taught about this in India. Not in that time. 6 years ago, 7 years ago, not happening. So I decided that I'll do the bioinformatics steps for it and, try to gauge how much of the challenges that are present for glioblastoma multiforme and how can I as a student try to solve even some of it if I can. There have been researchers who have been doing it for a very long period of time, but if I can make some significant impact, it might be good. Obviously, it didn't materialize that way, but what I learned from that was that in GBM, I'll cut it to the short form glioblastoma multiforme GBM, the cells of the tumor and after you cross your blood-brain barrier, the intratumoral and intertumoral cells are similar yet different. So they are in an immune suppressed environment. And this is what we call in scientific terms intratumoral and intertumoral heterogeneity. So they are different yet similar. And if you try to make a drug delivery target for that particular stem cell niche in that tumor, chances are that it will fail because it will attack the normal cells in the brain also once it crosses the blood brain barrier. So you have to be very careful while designing the drug, while analyzing all of those pathways which are present in your blood-brain barrier and inside those tumors. So there are some pathways which are have been focused more in the research. So there is the PI3K pathway, RB pathway, P53, as we all know, the tumor regulation. So all these pathways have to be accounted for. So this I was, introduced to, and I utilized a major part of Galaxy software, if you've heard of it. It's an open source data platform where you can do data intensive research, and then MATLAB for analyzing graphs and plots and all of that, heat maps. And then for pathway analysis, I was using Cytoscape, Reactome pathways, KEGG, Panther, David, all of these, so that I could get an idea of how something might happen. But of course, 3 months time is very less. It's good from a student's perspective that, wow, you're doing all of this. But for a researcher, it's nothing. People spend ages. So that was how I was, introduced to bioinformatics, proper bioinformatics, that this is how things can progress. So yeah.
Okay, so moving on, at UZA, you expanded the diagnostic yield for congenital hearing loss. So, could you elaborate on how your work with haloplex gene panels and minion sequencing advanced the field?
Yeah, so it's Minion. It's not Minion.
Benign.
We call it Minion, but it's Minion.
Okay.
So yeah, Haloplex gene panel is basically what you call just targeted gene sequencing. We have whole genome sequencing, we have whole exome sequencing. Haloplex gene panel is a product from Agilent Diagnostics, okay, the company. So there are other products in the market like from Illumina and other companies as well. Haloplex, what it basically does is just targeted gene sequencing for various diseases. I was doing a project on hereditary hearing loss, so I was focusing on the hereditary hearing loss panel that was being used at the university's lab and also in the diagnostic lab. Now, you guys know about targeted gene sequencing, or should I elaborate on that?
You could
That's fine.
elaborate a bit, like
Okay.
if you.
Okay. Yeah. So targeted gene sequencing is basically where only specific genes or a set of region of interest of genes is, sequenced, and then the read depth for that is a lot more, say 50x, 30x, 20x, how much ever you want to go into your analysis part for the bioinformatic reads to come out. That is what is targeted gene sequencing. Instead of analyzing the whole exome sequence or the whole genome sequence, which is like trillions of base pairs, you just focus on those disease-causing variants or genes. And inside those, you get the variants which can be, pathogenic or likely pathogenic, benign, or variants of unknown significance. So that is what targeted gene panel sequencing is. So it just gives you a higher coverage in the region of interest. So next was Minion, right? Minion is, if you heard of long read sequencing, it's a tool for long read sequencing. But the advantage of Minion is that it is very small and it is portable. So you can easily carry it in your pocket. If you might have heard back in 2016, 2017, when Ebola outbreak happened in Africa, Minion flow cells were used. They were transported from America or somewhere and then taken by flight in an emergency to Africa. And then the sequencing happened for the Ebola virus, and Minion flow cells were used. It gave such a good accuracy to get the whole sequence of the genome for the Ebola outbreak, that scientists were able to use it on the field without having to have extensive diagnostic equipments. So the basic fundamental concept of that is, for long read sequencing, that's you use longer fragments rather than the short base pairs that are used for next generation sequencing. So the complex DNA regions, which you might have, suppose like highly repetitive sequences or pseudogenes, or you have some splice site variants that are in the intronic regions, but they cannot be analyzed. So long read sequencing in one go can give you all of that information. And then you can get an idea after doing the bioinformatic analysis with various other softwares that what is happening in those regions, and you can make therapies for that as and when needed. So for my work, I was in at Belgium. I was working with the hearing loss genes. So the genes which are majorly complicated for hearing loss, which have pseudogenes, are STRC and AutoA. So they have many numbers of pseudogenes or highly repetitive sequences, if you may. So these hi-fi read sequences or long read sequences could simplify the process for the underdiagnosis or the patients who are not getting diagnosed, but they do have a hereditary hearing loss condition. That can be done easily. So that was where the idea for MinION sequencing was.
That's really interesting. So, uh, with your current role at the Gene Box, what excites you the most about the intersection of, say, pharmacogenomics, clinical genomics, and epigenetics in personalized medicine?
Okay. So do we know what pharmacogenomics is? Do you have a vague idea?
A little idea, a vague idea, yeah.
Okay, tell me your vague idea. What is it?
I'm blank.
No, it's okay. No pressure.
So I
Like,
think from what we've learned throughout our course is that it's something to do with the difference in like everyone has different polymorphisms in the
Mhm.
population. So that changes the ability of each individual to metabolize a drug. So there's differential metabolism. Something
Yes.
like that we've learned.
Yeah. No, that's spot on.
Yes.
Very good. So we all have different SNPs, as you said, and they can help us be categorized into for different drug types. I'm talking about we know about this CYP cytochrome 450 pathway, right? So it can help divide all of the drugs into 13 to 15 major genes. So it can be CYP2D6, CYP3A5, CYP2C19, DPYD for cancer. I'm saying cancer because DPYD is very essential for chemotherapy. If your body will have a positive or negative reaction to chemotherapy, or how much of an adverse reaction you might have with chemotherapy radiation going into you, DPYD can talk about that. Like if you have your pharmacogenetic test done, it will give you a list of comprehensive drugs. If you are a poor metabolizer for that, if you are an intermediate metabolizer for that, or an ultra rapid metabolizer, so you need very high doses because your body metabolizes that drug very quickly, ultra rapid. If you have a normal metabolism, then you need the standard drug dose. All of these are provided in CIPIC, DPWG, FDA guidelines. So there are databases, PharmGKB, PharmVar, which can be used for pharmacogenomics. So people are not aware that these things are in the market already. The GeneBox is doing it, MedGenome is doing it, MacMyGenome is doing it, Stand is doing it, but people are not aware. So that's one of the, I think the best things that can come out of this newsletter is that people should become aware that there are such things existing. If you have a loved one who is constantly falling sick due to, having too many medications, there is a term for that. It's polypharmacy. That, you are eating 7, 8 medicines at one time. Mostly happens in older people, our grandparents. So they can get their genetic test, pharmacogenetic test done and see what their effect of every drug or their dose on the system is. And with epigenetics and clinical genomics is very straightforward. You get a patient sample, you have to analyze whether they have cancer or whether they have any hereditary, condition, or they have any other rare disease, and you have to analyze the variants and then give them out a report stating that this is so and so. You have to, take care, and if there is any treatment available, you can refer them to the specialists in the hospital so they can go and talk with them. So my company in general just talks, we are a B2B company. We just talk one company to other or one hospital to our company. We don't directly deal with the patients. I'm just generating the reports or the back end of everything. The main work is being done by the doctors and the genetic counselors who are actually talking to the patients, actually making them aware of their risks. We are behind the scenes doing all of this. And for epigenetics, I would say because we have, such a deeply intricate relation with our lifestyle, our diet, our environmental factors. Everything affects our epigenetics is basically that. It's a regulation switch on off. क्या हो रहा है हमारे जीन्स अगर स्मोक कर रहे हो तो कोई पाथवे ऑन एक्टिवेट होकर उसका जीन एक्टिवेट होकर तुम्हें कैंसर का रिस्क हो सकता है नहीं भी हो सकता है अगर तुम्हारा जेनेटिक मेकअप अच्छा है तो इट ऑल डिपेंड्स ऑन हाउ यू आर टेकिंग इट राइट तुम्हारी बॉडी कैसे ले रही है so we just focus on making the quality of life better for the patient so that they can get that overview analysis for nutrition also from their reports. That's what we are trying to do. My founder is a visionary in that terms. Dr. Prana, not doctor, sorry, just Pranavanam. Yeah.
Moving on to our next question, what exactly motivated you to launch Genome Revelations, and how do you envision its role in increasing public awareness of genetics and genetic testing?
Okay. So when I came back and I had a fair bit of experience under my belt, and I was like, लोगों को पता ही नहीं है जेनेटिक्स के बारे में डीएनए पता है डीएनए इज द ब्लूप्रिंट ऑफ ऑल थिंग्स पता है सबके अंदर डीएनए होता है पता है डीएनए करता क्या है एक्चुअली तुम्हारी बॉडी में अपार्ट फ्रॉम डीएनए बीइंग डीएनए व्हाट इज इट दैट वी नो अबाउट डीएनए डू पीपल नो डू वी इवन हैव कन्वर्सेशंस विद आवर पेरेंट्स अबाउट डीएनए और एनीथिंग नो राइट सेक्स एड तो दूर की बात है डीएनए के बारे में बात नहीं हो रही है इधर पे दिखता नहीं है तो राइट तो I was like, there needs to be something, tangible that is giving free access to people so that they can have certain amount of genetic literacy. So I tried to always come down to the complexity of the research papers जो भी हम पढ़ते हैं इतने सारे to make our thesis, to do this work, that work. What is the common man getting from that? Nothing. If they go and read it, they are not going to understand. So my whole, the gap analysis was just that I want to be able to reach the masses by writing scientific articles which are so simplified in language and particular to genetics and genomics. So that even if someone comes on my website and goes through polypharmacy in the elderly and how PGX can help or what is genetics, what is PCR, everyone came to know about PCR during COVID. उसके पहले किसी को पता था? नहीं. अभी सबको बोलो पीसीआर हाँ पॉलीमरेज चेन रिएक्शन वो कोविड के टाइम पे होता था हमारा टेस्ट बट व्हाई आरंट वी टॉकिंग अबाउट द अदर थिंग्स इन जेनेटिक्स लैक्टोज इंटॉलरेंस है 90% ऑफ द पॉपुलेशन लैक्टोज इंटॉलरेंट है बट यू आर स्टिल ड्रिंकिंग मिल्क टिल अ सर्टेन एज बिकॉज पेरेंट्स फोर्स यू टू और समथिंग लाइक दैट बट तुम्हारे बॉडी डाइजेस्ट नहीं कर पा रही यू हैव एक्ने इश्यूज यू हैव गट इश्यूज यू हैव अदर इश्यूज यू स्टॉप ड्रिंकिंग मिल्क तुम्हारे आधे से ज्यादा इशू खत्म हो जाते हैं ग्लूटेन इंटॉलरेंस है क्यों है किसी को पता नहीं है जेनेटिक्स बात करो मेरा वही है कि देयर शुड बी कन्वर्सेशन and I try to write in a way that is clear and engaging enough for the audience so that they can be educated and also start a dialogue with their families so that they can have a happy discussion कि okay मम्मी ऐसा है मैं नहीं पी सकता और दूध मैंने ऐसे ही कन्विंस किया था मम्मी को अपने आई वॉज लैक्टोज इंटॉलरेंट एक्नी हो के मेरी स्किन खराब हो गई थी आई स्टॉप ड्रिंकिंग मिल्क 2 years में एक्नी क्लियर्ड मम्मी को समझाना कौन जाएगा नहीं इसलिए करना पड़ता है so that is what my main goal was that I want to bring it on or to one platform and then give them free access to read about genetic information and genomics also.
So building on what you said and like what you just mentioned, genetic information can sometimes be very overwhelming for non-specialists. So what strategies do you use to simplify complex topics while ensuring that the accuracy in your public outreach initiatives?
So I'll go back to my time in Govandi. So when I was doing that, as I said, the people were not from a highly educated background, right? मराठी में समझा रही हूँ, विजुअल्स दे दे के समझा रही हूँ कि ये आपका डीएनए होता है, ये आपका ऐसे रिकॉम्बिनेशन हुआ, ये फिर आपके बच्चे में ऐसे पेडिग्री चार्ट को समझा समझा के बनाया है, मराठी में समझाया है, हिंदी में समझाया है ताकि उनको समझ आए. So that is all a part of your outreach only. Dumbing down the complex information into a way that it is understood by the layman or the common man. So you can use visuals, you can use, experiences, personal experiences. जैसे मैंने अभी बोला वो lactose intolerance वाला. या फिर polypharmacy के लिए I've written an article on my website. My grandmother had a stroke. So when she had the first stroke, I was in Glasgow. And I did not know, but she relapsed in the next year also. And then subsequently 4 other strokes in the next years. And तब तक जब मैं वापस आई, I went and asked the doctors, why is this happening? मतलब क्यों इतनी सारी दवाइयां उनकी चल रही हैं? Why is she still relapsing on all of these medicines? And after every attack, she was getting worse. It's not like she was becoming better. In the first stroke, she lost all her speech and was paralyzed from one side. And she's still alive, but she's still having attacks. The most recent one was 3 months ago. So I asked the doctors why. They're like, ma'am, medicines react कर रही हैं. तो मैंने बोला कौन सी मेडिसिन रिएक्ट कर रही है, कैसे पता चलेगा? नहीं पता उनको. So awareness again, the awareness needs to come to the healthcare practitioners, the people who are doing the tests. हम हॉस्पिटल में जाके बस हाँ भाई डॉक्टर ने ये टेस्ट बोले हैं कर लो. Do we go and check why these tests need to be done? Mostly we don't. We trust the doctor's judgment. I'm not saying that's bad. The doctor has studied. He needs to, be well versed with it. But there needs to be some check on our side as well, that why are we doing what we are doing? So my aim is just ki on the website or through when I'm talking to people or giving webinars and stuff, when I have question answer sessions at the end, try to make them understand that even though this genetics is a mystery, or it's a very complex topic for some people, and there are many scientific aspects that cannot be understood in one go. I completely understand that. But the emotional burden that we feel from that stems from our genetic makeup, that is very much a lot of times overlooked. So we need to be ensuring a balance, that there is a comprehensive understanding between people, the doctors. There should be some level of, questioning also from the patient side, that why am I being told to do this test? So it's just the complexity into simplicity. That's what my major aim for my public outreach initiatives is like.
Very insightful. So, as we have been talking about it, epigenetics is gaining a lot of significant attention. So, in your experience, how do epigenetic modifications influence disease progression, and how are they shaping the current therapeutic approaches?
Okay, so epigenetic modifications, as I've already said, they are basically DNA methylation, histone modifications, chromatin remodeling, whatever happens, scientific terms, bade bade shabd. So now how they play a crucial role in disease progression. You want a scientific answer ki layman mein.
Layman. I'll prefer layman.
So our phenotype is affected by all the things around us, and our genetics also. Our genotype also affects our phenotype. Our lifestyle affects phenotype. Our diet and sleep and everything else also affects our phenotype. So what these epigenetic modifications do is they give us significant potential for clinical applications in terms of therapy, because they are reversible damage. Epigenetics, as I said, is an on-off switch. It gives us a reversible damage. Genetic में एक बार mutation हो गई, तुम्हारा risk factor अगर है वो pathogenic and you do not take care of yourself, you are more likely to get that disease. I'm not saying वो disease होके ही रहेगा, but epigenetics से अगर शायद तुमने manage कर लिया, if your sleep cycle is good, you sleep 7 to 8 hours, your diet is good, and you still have a type 2 diabetes risk and you are not overweight, you will not become type 2 diabetic. right? Type 1 diabetes is different, that is juvenile. But type 2 diabetes can be prevented with lifestyle modification. That is what the doctors also say. कोलेस्ट्रॉल हाई है, ट्राइग्लिसराइड हाई है, एक्सरसाइज करो, टाइम पे सो, टाइम पे खाओ, अपनी डाइट कंट्रोल में करो, फ्राइड फूड कम करो. This is all epigenetic modification that is happening on a genetic level, right? Sorry, not genetic level, in our body. So all of these affect you. So, but in terms of bioinformatics advancements that have been happening, or gene editing, or high throughput sequencing approaches that have accelerated in these past few years, they do give us a lot of new opportunities and possibilities to treat epigenetics, to treat diseases in the way of epigenetics. Although a lot of challenges and major research is needed for this, the deeper mechanism for the understanding कि वो कहीं ऑफ टारगेट पे जाके तो नहीं इफेक्ट कर देगा एपिजेनेटिक ड्रग अगर बना भी दी, जिसके लिए इंटेंट की है, अगर वो स्विच ऑन हो गया और उसकी वजह से कोई और ऑफ हो गया तो lot of research is needed. So it's not like it is going to happen soon enough, but I'll just say that there is significant research happening in that area also for metabolic diseases, cancer disease, and others for that matter. So it's a great initiative, or promising area that can be explored much further in detail by researchers.
So, given your work in pharmacogenomics, how do you see genetic testing influencing treatment strategies in the next decade? And are there any specific challenges that need to be addressed?
Next decade, देखो मुझे नहीं पता क्या होने वाला है. मुझे पिछले साल तक ये भी नहीं पता था कि हमारा 10,000 जीनोम्स वाला प्रोजेक्ट कंप्लीट होके लाइव हो जाएगा. मोदी जी ने जो इस ईयर हमें गुड न्यूज दी है दैट आवर database is available to the public also. So things are happening in the background a lot. I can't comment on that. Too much is happening on the global scale. For in the whole field of bioinformatics, genetics, biotechnology as a whole, lot of things are happening. And but I would just like to say कि अभी जो India and global scale में we have started using NGS for cancer diagnosis, or NGS मतलब not whole exome sequencing ज़्यादा हो रहा है, whole genome sequencing से थोड़ा कम, but the cost of it is falling down. Targeted therapy gene panels are even more prevalent than whole exome sequencing, and they are cost effective also. So I understand why that is the case. So a lot is possible in the coming decade with the advent of AI. Also, a lot of data that is being generated can be analyzed in minutes or hours, which was going to take humans अगर months लगने वाले थे या 2 हफ्ते लगने वाले थे, वो AI फिर भी 1 दिन में या 2 दिन में करके तुम्हें दे सकता है. So I'm not saying AI will take over, but AI will simplify a lot of things that are possible in the field of bioinfo at least, because at the end you need your human awareness to decide if this variant is going to give you a pathogenic result or not. You need that. So with more people understanding about genetic testing and being aware about genetic testing, healthcare providers themselves telling people that genetic testing is available, personalized treatment or pharmacogenetic testing that can give you a better control over your own health, that is very important. And as we start talking and drug optimization happens, better drugs come into the market, it will all converge into being a disease prevention kind of strategy, if you may. But again, the challenges are accessibility. हम access नहीं कर पाते हैं अभी भी चीजों को. Genetic testing, pharmacogenetic testing, all of these are very highly priced, I'll say. They are not included in health insurance plans. तो वो एक चीज that needs to be done on a global scale कि health insurance में वो माना जाना चाहिए कि इसका reimbursement होएगा. हमें इंडिया में तो नॉर्मल अपने हॉस्पिटल एडमिशन के reimbursement नहीं मिलते, उसके लिए भी इतना प्रॉब्लम होती है. So this needs to be taken care of. Equity, so equally distributed amongst all stratas of society. It's not like 5,000 का टेस्ट है, सिर्फ अमीर लोग या मिडिल क्लास लोग ही कर पा रहे हैं, गरीबों के पास कोई ऑप्शन नहीं बचा. नहीं, they need to get it done. Now how that happens is another problem which I don't want to get onto, but it needs to be taken care of. Then privacy concerns regarding the data. What uh, the amount of data that is being generated, how do we decipher which is good, which is bad, which can be shared with another organization or another researcher? Is it openly available? Is it like you have to take permissions from somewhere to get that data and then use it further? Usko bhi tum publish kar paoge ki nahi us hisab se? Privacy concerns. So that is all needed to be taken care of.
So, ma'am, you are someone who is deeply involved in research and public outreach. So, what are the gaps that you see in genetic literacy among the general public and also the healthcare professionals? And how can we bridge these gaps?
So as I said, healthcare practitioners, first of all, and the people both need to be aware that these things exist, and they need to be willing to go out of their way to learn also. Because जब तक हमारे खुद के अंदर वो क्यूरियोसिटी नहीं आएगी कि हमें सीखना है, तो मार्केट कहाँ से बनेगी, है ना? जकूजी सबको पता है, सबको पता है ना जकूजी. जकूजी ब्रांड है, इट्स अ बाथटब ना एट द एंड ऑफ द डे. क्यों बोलते हैं सब जकूजी जकूजी जकूजी? नो, इट्स अ सोना आल्सो इज अ जकूजी. उधर भी तुम बैठ के वो क्या हॉट वाटर बात ले रहे हो या जो भी कर रहे हो, बट यू से हाँ जकूजी में जा रहा हूँ. घर पे बोलते हो तुम बाथटब में, बाथटब बोलते हो. बाहर जाओगे जकूजी बोलते हो. ऐसे ही जेनेटिक टेस्टिंग के लिए तुम्हें बोलना पड़ेगा ना कि मुझे चाहिए. You need to be aware. Doctors, if they prescribe you, you should be able to tell them कि हाँ ठीक है, मुझे ये वाला ऐसे ऐसे चाहिए. हमारे पास वो क्यूरियोसिटी ही नहीं है जानने की. It all comes down again to that. It might seem like I'm reiterating the same point over and over again, but it is what it is, that the market won't grow. How did smartphones come into picture? Do we know? पहले वो नोकिया के साथ सारे खुश थे ना, छोटे वाले. फिर वो फ्लिप फोन आए, फिर वो स्मार्टफोन का छोटा वाला वर्जन आया, इतने इतने स्क्रीन वाला. अभी 6.97 इंच स्क्रीन पे पहुंच गए हम लोग. आईपैड आ गए हैं, 52-55 इंच स्क्रीन आ गई है. Someone innovated on that technology, right? Then they generated a demand for that कि ये तुम्हारे पास होना चाहिए. नहीं होएगा तो तुम्हारी लाइफ नहीं, तुम कुछ नहीं कर रहे लाइफ में. बट हम जेनेटिक्स के लिए ऐसे बात करते हैं नहीं. हमारे डायग्नोस्टिक टेस्ट, हम ब्लड टेस्ट करवाने से डरते हैं लोग कि हाय मेरे टेस्ट में कुछ गलत आ गया तो पता नहीं क्या हो जाएगा मेरे साथ. ऐसे नहीं चलेगा. We need to have open conversations. Education and communication is the most important, thing to bridge these gaps of research and innovation. So I'm just saying कि there should be social media campaigns. Instead of watching reels on dancing and stuff so much, people should also try to get into reels of doctors making good content, or students like you who should be making good content, professionals like me. But I know, will it do well on the reel segment? No. I have an Instagram page for Genome Revelations. I barely get any clicks. No one is interested in reading about that. No one. Because उनको चाहिए क्या कि brain rot. So there needs to be conversation happening and equipping clinicians with those tools to apply that genetic information that they get. So there needs to be constant collaborative efforts between geneticists, clinicians, educators, the government, the common people. There needs to be an improvement in genetic literacy. That is what is needed.
All of that was so impactful and insightful. And to move on to our last question of segment one,
Yeah.
and to conclude all that you said, what are your long term goals for advancing the fields of medical genetics and epigenetics? both in your research and through your initiatives like Genome Revelations?
Okay. So I am right now, I'll be very frank, I'm not more of a research person. I am more of a development person. Like I'm developing products that can impact the market. So all of that research that is being done, I have the knowledge to understand that and create it into a product which will help the consumer end. So you can get a DNA fitness report also. You can get a nutrition report. You can get your pharmacogenetic report, gut microbiome report. All of these are being done by my company and other companies like Strand and others as well. So that is where I'm seeing this. There needs to be, first of all, communication, collaboration, and consumer and market. वरना एडवांसमेंट नहीं होने वाली है. There needs to be empowerment to the public and the general masses in terms of genetic literacy for them to be accessible, to be able to make complex decisions regarding their own health and their genetic, and be able to understand what decisions they are making. That is what my advancing of this field means, that I want better health outcomes for all individuals across all strata of society based on their unique genetic profiles and epigenetic profiles and pharmacogenetic profiles and Jodi. But you need to be able to tailor your own medicine. It can't be one size fits all. There needs to be a conversation going, and there needs to be an active role from everyone included.
Okay, so that marks an end to our first segment. But
I
second,
actually wanted to ask a small question. Ma'am, so do you think that pharmacogenetic testing will play a role in personalized medicine in the future? Like even for cancer or any other disease? Like, will it play some role?
Yes, it definitely will. So, as I gave an example of DPYD gene, right? So, if you are a high metabolizer for DPYD gene, and if you're taking a chemotherapy drug during your cancer treatment, and suppose it is stage 1, stage 2 cancer, मैं higher stages पे नहीं जा रही हूँ, just suppose कि उतना कम है, and आपने treatment ली, but आप higher metabolizer हो, और आपको कीमोथेरेपी की ड्रग वो लोग स्टैंडर्ड डोज के हिसाब से दे रहे हैं, तो आपका सिस्टम से वो जल्दी निकल जाएगा, आपको वो उतना इफेक्ट ही नहीं करेगा कीमोथेरेपी का ड्रग. नाउ सपोज केस एकदम उल्टा है और आप स्लो मेटाबोलाइजर हो, पुअर मेटाबोलाइजर हो, एंड आपको नॉर्मल डोज दे रहे हैं, so there is a higher chance of you having serious side effects. You will vomit more, you will be hospitalized or feeling weakness and fatigue more after your chemotherapy. One session is done. After multiple sessions तो बहुत ज्यादा हालत खराब हो सकती है, depending on your metabolic status. So definitely it will play a role in terms of personalized medicine. Yes.
Okay, ma'am. Thank you so much. You can go ahead,
any
Vibha.
questions?
Yeah, I wanted to ask you a question. So,
Yeah.
will this also help in like the toxicity studies that we do? Because I think since people are given medicines and they're not able to metabolize it, that is why there are like so many toxicity issues that come up. So then
Yeah.
will this also help in like, like helping
Mm,
that segment of
yeah.
pharmacology?
So
Yeah.
now this comes under molecular studies of the drug that is being
Right.
made. That
Right.
is different.
That's a
That
different,
what
yeah,
research
right.
has to happen is very different.
Right.
Pharmacogenetic testing can't go into toxicity,
Right.
but
Right.
definitely it will give an idea that for this person this drug is toxic.
Right. Understood.
उसकी वजह से ड्रग नहीं चेंज होने वाली है,
Right.
ड्रग
Right.
का
Right.
डोजेज कॉन्सेंट्रेशन चेंज होने
Right.
वाला
For
है जो
that
तुम्हें मिलता
particular
है
person.
for
Understood.
that particular
Got
person.
it.
So if say Harsh is normal metabolizer and उसके लिए
Right.
standard drug dosage for Dolo is 10 mg,
Right.
हम कितना खाते हैं 650 mg या डोलो 600 650
600, yeah. Yeah.
या 600, और
Right.
इसका ड्रग डोजेज है सपोज 10, इतना कम नहीं होगा, मैं सिर्फ बोल रही हूँ, इसका
Right.
10 है और तुम्हारा है सपोज 500
Right.
एंड विभा का है 250 250, तो इट विल नॉट अफेक्ट ना, उसके सिस्टम से वो
Right.
निकलेगी ही नहीं डोलो,
Right.
बहुत टाइम लग जाएगा.
Right.
विभा के सिस्टम से ऐसे 4-5 घंटे में जो शेल्फ लाइफ है वो ड्रग की गई, for you, sorry, I said for you it's 500, right? So तुम्हारे system से ज़्यादा जल्दी जाएगी.
Yeah,
Vibha ka it will be moderate. She
somewhere
will take
midway.
7,
Yeah,
8 hours to metabolize that drug, and then she might,
right,
feel okay. So it will depend the drug dosage concentration for you. Toxicity is different. वो animal models में जो test होगा, वो सब drug
right,
के different molecular docking करके वो सब अलग चीज है.
right. Right. Thank you.
So now this marks the end to our first segment. Ma'am, your expertise on anything, everything that you've spoken about today helped me a lot because let me tell you, your lingo, the way you have broken it down, has helped me personally. So it was very
Great,
nice.
good to know that.
And like I personally have a difficulty with like huge terminologies, and you have
Uh,
effectively broken them down today. So which
okay,
means if I can understand, I'm pretty sure others can. And yeah, that marks an end to our interview. Thank you so much, and
thank you
to
all so
all of
much.
our viewers, readers watching this, kindly like, share, subscribe. Please check out ma'am's channel. Ma'am, would you like to plug in your LinkedIn and everything?
Okay, so my LinkedIn is Sohni Tagore. You can just find me. Sohni is like typical Punjabi spelling, Sohni. I'm a Punjabi. My surname confuses people. Tagore, I'm not Bengali. But I'm born and brought up in Mumbai. So I consider myself Mumbaikar. But yeah, my name is Sohni Tagore. For my LinkedIn, Sohni Tagore as well. Facebook, Sohni Tagore. Instagram is sony.tagore. And if you would like to go to my blog, it is www.genomerevelations.com.
Okay. I think
So
that's
thank you
it.
so much.
Thank you so much.